So hello everyone and a very warm welcome to this FLV Global interview. My name is Colin, so thank you all for joining us via YouTube or podcast. Today our special guest is Dr. Vera Gorbunova. Now, apart from being on GenFlow's Scientific Advisory Board, Vera is a professor of biology at the University of Rochester, New York State, and the co-director of the Rochester Aging Research Center. Her research is focused on understanding the mechanisms of longevity, the studies of exceptionally long-lived mammals, and the subsequent genome stability. And for those of you that are obviously interested in GenFlow Biosciences, they're a UK-based biotech company aiming to deliver therapeutics that potentially halt or slow the aging process in humans and man's best friend, the dog. The company's lead compound works through the delivery of a centenarian variant of the CERT6 gene and has yielded very promising preclinical trials, but more of that later. So, all the way from New York State in America, welcome and good morning, Dr. Vera Gorbunova.
Oh, good morning.
So now, before we start, I believe you just taken your dog, your beloved dog, Wolfie, to the vet. How is he?
Well, he's doing better. He received medication, and Wolfie is 12 years old, so he's facing the same problem people face when they get older, so he needs more medical care.
Is he going to be part of the GenFlow trials, do you think?
Yes. I start giving him some medicine already that, have some indications for having pro-longevity effect, so he is my patient number one.
Excellent. Well, look, thank you very much for joining me today. I know you're very busy and you've got another meeting very shortly, so I do appreciate your sparing time out of your busy schedule. Now, I've just mentioned you currently work at the University of Rochester, but you originally studied in Russia at the St. Petersburg State University. So before we get into the heart of our discussion, would you please give our viewers a quick resume of your journey from Russia to America and your working life to date?
Well, I was born in St. Petersburg in Russia, and in the family of two physicists, theoretical physicists. And I was interested in science early on, but physics wasn't my thing. I wanted to study something that is closer to people and to living things. And I became interested in biology very early, but I wasn't sure which area of biology, so I started my education at St. Petersburg State University, where I completed my bachelor's degree. From there, I continued to my master's and PhD degrees at the Weizmann Institute of Science in Israel, which is a great institution. I was working on questions related more to genome stability at the time, although I was interested in aging research, but there was no one working on it at the time, so I had to postpone it a little bit. And then for my postdoctoral studies, I went to United States, and at that point, I was certain I want to be in aging research. And I started aging research in 2000 and been working in this area since.
Now, during my deep dive investigation on Project Vera, I saw that you worked for many years in cancer research with some quite remarkable results. I think in 2009, you published research that showed that a hairless underground rodent, aka the naked mole rat, expressed a gene called P16 which prevented cancer tumours from developing. Was this something you suspected was the case with these mammals, or were you blown away by the results?
This is another area of my research where we study long-lived mammals, and the idea is that once we understand the mechanisms for their natural longevity that they acquired in the process of evolution, and once we understand those mechanisms, then we can apply them to extend lifespan in people. And to live for a long time, you have to stay cancer-free. So those two fields of research, to me, are very closely connected. Cancer resistance has to come together with longevity, and many of the treatments we consider for longevity, they also can protect from cancer, as any age-related disease.
Yeah, so was it a natural progression to migrate from cancer research into longevity, or are they inextricably linked?
They are linked. I always consider myself aging researcher, but we just often find that, we are working with cancer because if we have this remarkable animal, for example, that naked mole rats, they live up to 40 years and they are same size as a mouse, and they don't get cancer. and they also don't get other age-related diseases. So which one comes first? This is all linked, and often the mechanisms are common, and we can target them with the same interventions.
Oh, that's interesting because I was when I think well, my prospect is or my perception is that when we look at life as a whole, biologically speaking, it sort of seems to revolve around reproduction and not living forever. It seems we're destined to be finite creatures. So I think before we talk about longevity, I'd like you to tell us what aging is, please, in that case, and importantly, what Sirtuin-6's role is in that process.
Well, there are many definitions of aging, but perhaps the simplest one that reflects, getting closer to the state of mortality. So when you talk about a population, so you see that mortality increases over time in a population. If you take a cohort of, animals or people of the same age, so there is a increased mortality over time, which is the result of aging process. Now, if we're thinking more about individual aging, so that would be a process of continuous deterioration that occurs over time. And it may not necessarily be linked to disease, because if you think about, for example, Olympic records, they are mostly held by people in their 20s, and maybe people in their 40s are not really maybe very healthy, but still they don't run as fast as a 20-year-old. So there is a process that is not linked to disease, but over time it makes us more susceptible to disease. So that's when age-related diseases start to manifest as well.
And Sirtuin-6's role in this process?
Well, yeah, so Sirtuin-6 is an absolutely fascinating gene and protein. It may be linked to those very fundamental mechanisms of aging. Whatever is the cause, the driver of aging. Because you can see many signs of aging, gray hair, maybe developing age-related disease. But like, these may be consequences, like what's the cause? And there are many opinions on the subject, and this is a very rapidly developing area of research. But what we, currently the most common thinking, and that's what I personally believe, is that the underlying cause is a deterioration of genome and epigenome. Because in the cell, if you think about living cell, many parts of it can be replaced and recycled, like proteins can be recycled, you can make new ones, lipids as well. But DNA is one part, like once something is wrong with it, it's very difficult or impossible to replace or recycle DNA. And the SIRT6's job is maintaining DNA in good order, in terms of not having mutations, because it helps DNA repair, and also keeping it organized well, because DNA is a very long molecule, and it can be very tangled, right, if it's not properly arranged in a way that it's accessible to enzymes when necessary. So SIRT6 is epigenetic enzyme, so it's important for packaging DNA properly within the cell. And as we get older, DNA gets disorganized and it accumulates mutations. And SIRT6's job is still coming in and cleaning it up and arranging it back. So what happens over time, SIRT6 can't keep up anymore. And again, as you mentioned earlier, well, natural selection doesn't really work for longevity, it works for reproduction. So we have enough Sirtuin-6 to take us through our reproductive years. But afterwards, there is not much selection, there is not much need to keep it going the same way, so things start to deteriorate. And here, what's really fascinating was shown by experiments in mice. If you give them extra dose of SIRT6, extra copy of it, then they live longer, because now you kind of bypass what evolution allotted to mice. Now you give them more, and that allows them to live longer. And also, if you take out SIRT6, you mutate it, then you get very sick mice. Their genomes are very disorganized and they die prematurely.
So I've seen that the bowhead whale can live to about 210 years. Obviously, we don't live that long yet, but there are various places around the world where longevity rules supreme. I assume the centenarian variant of the CERT6 genome was found among these groups. What's the significance then of this variant, and how was it discovered?
Yeah, it's a very rare variant. It was discovered by sequencing genomes of centenarians, people who live to 100 and beyond, and then comparing, because of course when you look at human genomes, you can find many differences. We are not like inbred mice. We have many differences in the genome, but what we analyzed were the differences that would be more frequently found in centenarians than in controls of the same ethnic group, same population. And this variant of CERT6, we actually almost never see it in non-centenarians, but we found several cases in centenarians, so it was overrepresented in centenarians. And then when we engineered this gene to work in cultured cells and also in vitro, in test tubes, and tested its properties, it turned out that it's better at maintaining DNA. It's better at folding DNA in proper structure. It's better at mediating DNA repair. So all the good things that you'd want to have, but you have to be exceptionally lucky to have actual variant, because not every centenarian has it. Even among centenarians, it's fairly rare. So what we are now trying to do is develop strategies with GeneFlow that everybody could benefit from this variant. You wouldn't have to be an extremely lucky centenarian, but for all of us, we could benefit from this unique sequence.
So then is there a possibility of swapping these cells with non-centenarian humans? Eventually, is that the plan?
Yes, so the plan is to kind of deliver a copy of this gene to non-centenarian humans, and so we could express some of it and have some of this protein in our cells as well.
So after all this groundbreaking work, what are the expected benefits of developing a product using this CERT6 variant?
Well, we expect many benefits, but most importantly, we expect to slow down this physical deterioration, onset of age-related disease, because we want to keep people healthy for longer, and extending health span is the most important objective. It may also extend lifespan as well, because if people stay healthier, they will live longer as well. But the first goal is just to keep people healthy for longer.
Of course. Now, we've been big fans of Dr. Eric Lehr and GenFlow for quite a while. I remember he was very excited to get you onto GenFlow's scientific board. How did he tempt you to join GenFlow? Was it similar ethos to your research that excited you? Or did Eric tempt you with something juicier, like an endless supply of naked mole rats?
Yeah, well, I was quite excited about his approach. Well, it is extremely close to what we do, and I've been researching CERT6 for a long time. We are also researching chemical activators of CERT6, and this may be, sort of more conventional approach, because we are looking for chemical activators, some medicine you can take and just boost your CERT6 activity. But Eric's approach was very bold, to use gene therapy. And I like this idea because, often people shy away from gene therapy, but now, nowadays this is becoming reality. And this is why I was excited to get on board with this initiative.
Excellent. Well, look, I really know you're short of time because you've got a meeting. We're overdue already. But so I think it's a pretty good ending to our webcast today. I'm sorry it was so short, but we'll conclude the interview here if you're happy. So please keep us appraised of any new exciting findings, as I'd love to chat to you again, hopefully longer next time when I know you're always busy, but hopefully next time we can have a bit more of an in-depth chat. So I'll end with a quote from the annals of Star Trek and one of the greatest proponents of longevity, and the immortal words of Dr. Spock, Dr. Vera Gorbunova, Professor of Biology and member of GenFlow's scientific board. Live long and prosper.
Well, thank you very much, and we'll be happy to talk again.
If anyone has any further questions for GenFlow, please email them to our dedicated email address at genflow@firstlookventures.co.uk, and we'll get you an answer into Genflow's exclusive Telegram group as soon as possible. Thank you once again for chatting with me today and our community.